So Morgan and his students did a double heterozygous cross. Heterozygous for straight wing and black body, which is big S, small s, big B, small b, crossed with big S, big B, small b. Double heterozygous cross. They assumed they would get the classic 9:3:1 ratio. They did not. Here is what they actually observed. 44% had straight wing and black body. 15% had curved wing and black body. Another 15% had straight wing and gray body. And here was the kicker. 26% had curved wing, black body, had the recessive type. Now the curved wing and black body, or recessive type, is very different from the parents, as we can see. In fact, it has a special name. It's called the recombinant type. Now that's because the parents' genes recombine in a unique way to give traits not seen in the parents. Now the only way for this type to show up is from crossover in prophase during meiosis. Prophase 1 actually during meiosis. These results we see here, the numbers, show us that the genes for wings and body color are linked genes, meaning they're on the same chromosome. These results also show us how closely linked these genes are. No matter how many times we do this cross, we'll always get between 26 and 27% curved wing with black body. This consistent pattern is telling us something. It's telling us not only that these genes are on the same chromosome, it's telling us how far apart these genes are. It's telling us how to make a map to find these genes. Now just think about it. These genes are on the same chromosome. If they're far apart, it'll be very easy for them to separate during crossover. Therefore, the frequency of this recombinant would be very high. But if these genes are very close, then they would hardly ever separate. The frequency would be really low. In this case, the frequency of 26% shows us almost exactly how far apart these genes are, because the frequency is how often this offspring shows up, which is therefore how often these genes separate. Okay, let's see what we're going to learn next.